Welcome student in the 5th week of the SWAM course Anatomy of Flowering Plants, and in the first lecture we will be talking about plant cell structures. Now before this particular module we have discussed about the cell structures, and we have discussed how different groups of cells are giving rise to special type of tissue which has unique structure and they perform different functions, and how different types of tissues are also organized to give rise to tissue systems. We have talked about epidermal tissue system, then ground tissue system and vascular tissue system, and now we will discuss that how these different types of tissue systems are arranged to give rise to different organs of a plant, organs or different parts of a plant or different structures of a plant. So, in this module, we will be talking about the important parts of a plant. Mainly, we will be discussing about the stem, root and leaves. As you all know that a plant is divided into a root system and a shoot system. Okay. Okay. So, plant will have shoot and a root. Okay. So, in shoot also you will see various organs, like you will see leaves, you will see axillary buds, then flowers will be there. Okay. Leaves will be there, then buds will be there. Okay. Fruits will be there. And in root also you will see the main root, lateral roots. So, among them we will discuss the three important structures which are present in a plant. Now, we will start with the description of stem. Now, stems are typically they are above ground organs that grow towards light. Okay. Okay, so they are growing towards light, positively phototropic. Okay. Generally they are above ground, okay, but some exceptions you may find that you will find some underground stems also, but they are very rare and they appear in some sort of modifications. The basic function of stem is to provide support and help in conduction. It provides support to all the parts of the plant which is present above ground. And it helps in conduction of water and nutrients, okay, from the soil to the various parts of the stem like leaves and flowers, and then it also conduct food material to different parts of the plant. Another important role of stem is production of leaves and flowers. Okay, so other lateral branches comes out from the stem. It also typically contains apical and axillary bud. Now, at the tip you will find an apical bud, and you will also find axillary bud which are found in the axil of a leaf. So, this is apical bud and this is axillary bud. Apical bud increases the height of the plant, increases the height of the stem, and it contains apical meristem, the growing point, okay, which is responsible for increasing the length of the plant, as well as the axillary bud. It gives rise to lateral branches, as well as flowers also comes out from this axillary bud. Okay. And very important feature of stem is presence of nodes and internode. See, when you will observe stem, you will see that there are distinct regions, it is divided into distinct regions which is known as node, and between two node there will be internode. Okay. Now, this internode, the length of this internode can vary Like you would have seen bamboo tree, okay, it has very long stem and very long internodes. Sugarcane also you would have observed internodes. Then many other plants also, the length can vary. A very short internode could also be observed in rosette plant. Like you would have seen cabbage. In cabbage you would have seen the leaves are there, the green part, basically they are leaves, and these leaves are arranged on a stem which has very small internodes. So you can say that the stem is quite condensed and with very short internodes, and because of this whole structure becomes highly condensed and the sideways leaves are coming out which appears to be overlapping. So this internode size definitely varies in different plant species. Now the function of this stem is definitely it is for support and for conduction, but apart from that it serves also other function, like it stores food. Some of the stems can get modified and they perform the function of storage of food. It also performs photosynthesis. Now photosynthesis is basically the function of leaves, but stem also, when they are green, they contain chloroplast and they could also perform photosynthesis. It is also in many cases they are adapted even to perform photosynthesis. However, in many normal herbaceous species you would have seen that the stem appears green in color, and it is because of the presence of green color pigment in the parenchymatous cells, which now it is called as chlorenchyma, and they will perform photosynthesis. Many of the stems also get modified, and it may resemble other plant parts, and we may not be able to identify that it is actually a stem, but they are modified to perform various functions. Now the stem generally it is erect, okay, it is found upright above ground level, but many a times you could see that the stem is also lying on the ground, they are prostrate, okay, lying on the ground, like in grasses you would have seen like that. Now before moving on to the anatomical structure, let us just briefly try to understand what are the different modifications the stem could take place, so to perform different functions. Now the underground modifications, in which the stem is found underground and it is modified, like you have rhizome, you have bulb and comb. Some examples are there, like rhizome is an underground modification of stem where they are actually lying almost in a horizontal manner inside the soil, okay. An example is like ginger and turmeric. So this is actually a stem, but it doesn't look like stem, but this is a modification of stem, and they are found underground. You can see here some scar-like structures, so they are nodes, internodes, and scale-like leaves are there. Okay, scales are there. Leaf is not well modified and they are not performing all the functions of a particular leaf, but they are reduced to scaly like appearance that you would have seen also when you have taken when you are trying to peel off a ginger. You will see that some scale like papery things are coming out. So they are scale leaves and they will have nodes and internodes in them, and they are modified to store food. Similarly, tuber is there, which is also an underground modification, like potato, okay, it's a tuber. So it is also a modification of stem, which is basically modified to store food. We all know that the potato contain lot of starch, so they store starch. And in potato also you will find nodes and internodes, you will find some eye-like structure. Okay, this is basically, you can say it's a node. It contains a bud which can give rise to which can germinate and give rise to a plant. Similarly, bulb is there. You would all have observed an onion bulb. Okay, so onion, the part which we actually eat, they are fleshy leaves. Okay, these are basically leaves, and the stem is modified into a disc-like structure. It is highly reduced. It is modified into a disc-like structure, and from these stems, roots will come out. So it is also a type of modification. Similarly, comb is there, which is also an underground modification in which the stem is vertical in orientation, okay, underground it is vertical in orientation, and it is also to store food. Then we have subaerial modifications. Now subaerial modification means that they are almost running on the ground, okay, they are almost running on the ground or slightly below ground, okay. In this, see, you all would have seen grasses, okay, these grasses run almost parallel to the ground, and these grasses have actually stem. See, and these stems are horizontal, they are found on the ground. You will see distinct places which are nodes. From there some roots will come out, some aerial leaves will come out, okay. So these type of stem modification you see, and it is basically modified for faster propagation. You would have noticed that when grasses or a lawn, in a lawn when grasses are there and you cut the lawn, then also leaves can come out, or even in a very dry condition when you see that the almost that entire lawn has been dried up, but then also in the next season the leaves and the stems and all that plant comes out. It is because some portion of the stem or some portion of this plant is still present in the soil surface, and when a suitable environment is obtained, they will come out through vegetative propagation. That means they don't need flowers or seeds to propagate. They can even grow with the help of stem. See, that also we have discussed why they are doing so, because they have intercalary meristem, which was left behind when the apical meristem was growing. See, we have three types of meristem. So apical meristem was growing, and in between nodes and internodes, we have intercalary meristem. So these are the meristem which is responsible for vegetative propagation. Even in bamboo trees also you would have seen that intercalary meristem is becoming active, and it is because of that only that long internodes are found. In this also vegetative propagation occurs because of the presence of this intercalary meristem. Now in this also different forms are there. Runners are those which completely run parallel on the soil surface, okay, they are running parallel and they have nodes and internodes, okay. Then you have stolon, okay. In stolon what happens, there is a main plant, and from there some from the main axis a stem originates and it falls on the ground, and from there it will give rise to another plant. Like in strawberry, you can see here, this is the main plant, and from there some stems are coming out, and they will, as soon as they will touch the ground, from there roots will come out and new plants will come out, okay. Then you have suckers also. Suckers are those in which they come out from the below part of a stem. From an underground stem they will originate, and as soon they will reach the ground surface, they will form new plants. Offset are similar type, runner or sucker. However, it is found in aquatic plants, okay, like in Pistia or Eichhornia, okay. So all the this is an example of Pistia. We can see nodes are there and internode is there, they all are connected, and from nodes roots, adventitious roots are coming out, and then leaves are coming out, okay. So this is a modification which is basically meant for faster propagation. Then you have aerial modifications in which stem is modified to perform various functions apart from providing support and conduction, in which the stems could be modified in the form of tendrils. They also provide support. This part is there, okay, it comes out from the axillary bud. Now from axillary bud tendrils will come out, and they will also help in hanging the plant or help providing support to the plant. Similarly, thorns are there. Now this tendrils you would have seen in like pumpkin and cucumber, in grapes also some coiled structures are coming out, some delicate coiled structures are there which you would have seen in all these plants. Then thorns are there which are spiny structures, okay, they are coming out from the axillary bud, and they provide protection from grazing animals because they are pointed and they have spiny structures, so they can harm grazing animal when the grazing animal come to pluck the to eat the leaves. In Bougainvillea, you can find thorns are there, which are modification of stem. Okay, lemons also you will find. Similarly, bulbils are a modification of stem, which are modified for vegetative propagation. Okay, they will also come out in the form of rounded structure, and they are also modified axillary bud. They become fleshy, and they will detach from the plant, and then they will give rise to a new plant. It is also found in some plants. It's not, these are not things that they are common in every plant. Now, cladode and phylloclade are basically, they are flat stems, okay, which are green in color, and definitely they will perform photosynthesis. Many a time these words are interchangeable, but the thing is that they will be flat stems, they will be green in color to perform photosynthesis. That means they have been modified, and generally these structures you will find in arid environments where to prevent water loss, leaf is highly reduced, like they could be in the presence of modified in the form of spines. So the function of photosynthesis is now performed by stems. Like this is a cactus plant, where you can see the entire stem is modified into a flat fleshy structure, and leaves, these spines are actually leaves. Okay. Asparagus, this is asparagus. Okay, they are also modified leaf, they are also modified stems. You can see here they look like leaf, but they are modified branches and into a flat structure. Then herbaceous stem, we call a stem to be herbaceous or woody. Now we call it herbaceous when only primary growth is there. That means the stem is not very thick, not a very wide diameter, and only they have grown till primary growth stage. There is no secondary growth which is occurring in these stems. So they will be definitely short height, and they have weak stems, green stems, and mostly annuals or sometimes biennial plants could also be herbaceous in nature. Woody stems are definitely perennial. Okay, because they grow for a very long period of time, by many seasons, even decades, or even more than that. So in that stem, secondary growth occurs. Secondary growth occurs, and this will give rise to wood. That means the secondary tissue will be definitely, they will be more harder, they will be more stronger, and we call it as wood in a generalized term when it has a very big diameter and it is a very big stems are, broad stems are present. Now the secondary growth, it occurs from vascular cambium. Okay, so this all we have discussed, that vascular cambium will give rise to secondary growth, it will form secondary tissues, it will form secondary xylem, then secondary phloem. Okay, then in that also it will form different xylem tissues and different phloem tissues. Okay, so all these things, and this will be a massive structure. Now vascular cambium, we all know that during the growth of meristematic tissues, different layers of meristematic tissue was formed, and procambium was giving rise to vascular tissue. So procambium forms vascular cambium as well as primary xylem. It forms primary xylem, primary phloem and cambium, and this cambium will give rise to secondary xylem and secondary phloem. Dicot stems could be herbaceous or it could be woody also. Okay, that means generally in dicot stems secondary growth is there, but in some very like herbaceous species which are very small, very weak, in those species only sometimes secondary growth formation is not there, but otherwise in most of the cases Secondary growth occurs in dicot stem, but it is important to remember that in monocot stem, secondary growth is lacking. So, monocot stem does not show secondary growth, and you would have seen that these stems are quite weak, like wheat is there, rice, grasses. You all have observed these plants, and you would have seen that they have very weak stems, very thin, and they generally they do not grow to a very larger height. However, there are many cases, but that also we will discuss, that how strength is given to those tissue which grows to certain height. Now, this also we have discussed, that the stem is composed of 3 tissue, okay, dermal tissues. Like every stem will have an outer layer, okay, it will have an outer layer. So, that will be epidermis or dermal tissue, and that will be derived from procambium. Then they will have a ground meristem, means like cortex is there, pith is there, okay. So, all these things will be there. Then procambium means the one which will give rise to vascular tissue, okay. So, vascular bundles will be there. So, that will come out from the procambium. Okay, now coming to a typical dicot stem, okay, although lot of variations you will see, but let's try to understand what is the structure and organization of a typical dicot stem. So, definitely dicot stems are solid, okay, they are not hollow from the center, and the distinct regions are present in a dicot stem like epidermis, then cortex is also subdivided, then you will see pericycle, vascular bundle and pith. So, let us try to understand the structural organization of these tissues. Okay, so this is the structure of a typical dicot stem. Typical structure of a dicot stem, you will see trichomes here. Okay, on the outer side you will see hair-like structures, they are trichomes. And then you have hypodermis, okay, which is collenchymatous, okay. And then you will see cortex, general cortex is there. And then you have after cortex, the lower layer of cortex is known as endodermis. And then you have pericycle. Now in this case you will see pericycle is heterogeneous in nature. It is made up of sclerenchyma and parenchyma. Okay. Now the parenchymatous patch which is found around the vascular bundle is also known as bundle cap. Okay. And below that you will see vascular bundles which are arranged in the form of rings. So it is a very peculiar feature of dicot stem that vascular bundles are arranged in the form of ring. Then you have phloem, okay, present on the outer side, and then you have xylem, okay, which is present on the lower side, and in the center you have pith, okay, a large pith. Pith is generally large in dicot stem, okay. Now the cellular diagram also we can make. Suppose this is the outer epidermis, okay, and then we have collenchymatous layer. So they are thick wall cells, okay, thickening in the side walls. Then you have parenchyma, okay, which are loosely arranged cells, okay, lot of intercellular spaces are there. Then you have endodermis, okay, which are barrel shaped cells, and they are also known as starch sheath because of the presence of starch in that. So this is also important that in dicot stem the endodermis contains starch and it is also known as starch sheath. Then below that you have sclerenchyma patches, pericycle of sclerenchymatous patches. Okay. And then you have vascular bundle, you will have cambium, 1 or 2 layers. Actually cambium is single layer, but that all we will discuss, that sometimes it appears multiple layer. Okay. Then you have phloem, thin wall phloem cells are there. Okay. So this is phloem cells. Then you will have vascular bundles where the arrangement will be endarch. So this is the vessels, and in between you will have lot of other trachees and fibers as well as parenchyma. So this is metaxylem and this is protoxylem. And this condition is known as endarch. So this is also very important that in stem you have endarch condition, and in between you will have the pith cells, okay, they will also be parenchymatous in nature. So this is the pith, this is the collenchyma, okay, and this is the cortex, which is, you can say it is the hypodermis, okay. Now these are some of the structures, pictures taken for dicot stem. Here also you can see that the dicot stem is arranged in the form of vascular bundle, and this diagram also I have given you in previous module to identify the parts, where you can see that outer epidermis, then these are the vascular bundles, okay, outer sclerenchymatous patches there, then you have phloem, cambium and xylem, okay, in the center you have pith. This is also an image of a pea, which is also a dicot plant stem, where you can see slightly different, it's not circular, it has a slightly different shape, but it is also a dicot, where you can see these are also arranged in uniform pattern. Pith is also there in the center. When you will see structure under microscope, here also you can see that this is sclerenchymatous patch, then this is phloem, okay, then you will have cambium, and this is metaxylem, and this is protoxylem towards the center. These pink structures, because these are dead tissues, and we generally a stain is used known as a fluoroglucinol. Okay, this particular stain those cells which have lignin in them. So definitely dead cells like sclerenchymatous cell, trachees and vessels, they will stain pink because of this dye. This is also a structure of a dicot stem which has slightly different orientation, cucurbita stem, in which you can see two, but it is bicollateral. Cambium is present on the lower side also, and on the outer side also. Phloem is present on both the sides. So you will have a central xylem with two cambium and phloem on the outer side. Even in the cortex region also you can see some cells are sclerenchymatous, some cells are parenchymatous, and some are sclerenchymatous. So different orientation in the cortex also it is observed. This is also a section of a stem which you can see that a transverse section, that is TS, and longitudinal section is shown here. See, when you cut a stem, suppose this is a stem and you are cutting them in this plane, this is the TS section, and if you are cutting them from the radius, suppose you are dividing them, you are taking this particular whole section. So you, in this you will see radial arrangement of cells, like radially arrangement of trachees and vessel, phloem, parenchyma. So in this radial section also you can see that in transverse you have epidermis, then parenchyma, endodermis, xylem, and all those tissue. So here also you can see that how they are radially arranged. They are definitely, they are slightly elongated in radial orientation. Protoxylem will have spiral and annular vessels, whereas metaxylem will have pitted vessels. Okay, similarly phloem will also sieve tubes you will see. Okay, sieve tubes, these are all on the outer side companion cell. So in this way you will see the entire organization which is there in the dicot stem. Similarly, this primary xylem and primary phloem, so that all we have discussed, that protoxylem and protophlloem will be followed by metaxylem and metaphloem. That initially, first of all, protoxylem will be formed, and then metaxylem. So protoxylem will have annular thickening, whereas metaxylem will have more harder lignified structures. Similarly, in protophlloem also you will have sieve tube elements, but companion cells will not be there because still they are growing and elongating. So these type of structures you will observe. After secondary growth formation, vascular cambium will take over and form secondary xylem and secondary phloem. So all these structures are basically primary. See, don't get confused that they are anyhow secondary structures. These are all protoxylem, metaxylem, all are primary structures. So this was today about the structure of dicot stem. In the next lecture, we will discuss about monocot stem. So thank you for today.